video for today has to do with bacteriophages. So these are viruses that only infect bacteria. So in general, we have two different types of bacteriophages, two different general groupings. The first one is virulent bacteriophages, and then the second one is temperate bacteriophages. Virulent bacteriophages only have one sort of option when they enter a host cell. They are going to enter into the host cell, or rather their genetic information is they're going to turn it into a viral making factory. You can hear my dog drinking water right now. And then they are going to rupture that host cell and cause massive release of commonly hundreds of phages that can then go on and infect. So there's one option here. Following infection, you get destruction of the host cell. Okay. T4 is a bacteriophage that's been heavily studied, that's a virulent one. The other option are temperate phages. So our most well-studied example of a temperate bacteriophage is lambda phage. The temperate phages have two separate life cycles that they can undergo when they enter into a host cell. So one of these is the lytic cycle, and in the lytic cycle, what they're doing is they're acting just like a virulent phage. If they go into a host cell and they enter the lytic cycle, they turn it into a viral making factory, they lyse it, the host cell dies, hundreds of phages are released. But the other option is that they would enter what's called the lysogenic life cycle. And in the lysogenic life cycle, what happens is for a period of time, the viral genome gets incorporated into the host cell genome. And so here in red, we'll have my viral genome, and then in purple, we'll have my host cell genome. When you have a phage that is in the lysogenic life cycle, and the phage genome is incorporated into the host cell genome, the virus is now known as a prophage. The bacterial cell, on the other hand, is known as a lysogen. So if we were talking about the host cell. Okay. Now, the advantage to this, if you are the virus, is that every time that bacterial cell replicates its genome and its offspring replicate their genomes, the phage also gets replicated without having to do anything itself. The other advantage to this is it's a way for the phage to hide out when conditions are bad, when there aren't a large number of host cells around. This phage can persist as long as the lysogen and its offspring persist. Okay. Now, in the event that things get bad, the bacterial cells are under stress, especially stresses that trigger DNA damage, like UV light, for example, or particular mutagenic chemicals. Then this prophage can undergo a process called induction. And what happens during induction is the phage switches from the lysogenic to the lytic cycle. Okay? And so essentially the sort of way to think about this is that if things start to get rough and there's a chance that the host cell is going to die anyways, let's turn it into a phage making factory, get a couple hundred phages out of that, and then kill it ourselves. And so again, it's sort of a safety mechanism, a way for the phage to replicate even when conditions are bad. So two options: virulent, total host cell destruction. That's the only option. Temperate phages can go into a lysogenic life cycle where they can persist for generations of bacteria. Or they can enter into a lytic cycle where they act just like a virulent phage. The lysogenic phages eventually, when the host cell starts to get stressed, will switch to that lytic cycle and then again act just like a virulent phage.